This is where we left off, and what we're trying to figure out with this particular experiment is it the protein or is it the DNA? And we took the phage, which they knew was made up basically of protein and DNA, and made each portion radioactive. You don't have to know exactly how. One portion is going to have sulfur and one portion is going to have phosphate. And they actually weighted one. So the protein portion, which would be the capsule, remained very light. And the DNA portion with the phosphate, they actually put this pellet in. So that when they centrifuged it, when they and they injected this into E. coli cells, just like I said, when they and then they grew both of these and injected them into cells. Then they took the cells and centrifuged them. We know we've seen this particular technique before, where you're going to centrifuge and you're going to actually centrifuge based on weight. So the protein would actually be much lighter than the DNA portion, so they could actually look at it under the microscope. And then they also could test its radioactivity. They found that the portion that was in the E. coli cell was the DNA, and so they concluded that it was the DNA that provides the genetic information. This was kind of the death knell to that particular controversy. Okay, so they knew it was the DNA, and the next gentleman who came along in the 50s, we're working our way towards around the 53 time when Watson and Crick made their discovery. That was the beginning of this particular chapter. But this man named Chargaff, what he did was then looked at the DNA and isolated the 4 different nucleotide bases. And what he found was, from species to species, the amounts of each were characteristically in a very common ratio from species to species. Now, please understand that a fruit fly has a different DNA makeup than ourselves, but what you have here is, so E. coli, you have about the same amount of adenine as cytosine. I mean, about the same amount of adenine as cytosine. Guanine and, I'm sorry, I'm all messed up. Adenine as thymine and guanine as cytosine. And if you look at wheat, adenine, thymine, sea urchin, adenine, thymine, and those happen to be end up being the pairs that always pair up. And then if you look down the row in terms of guanine and cytosine, very similar numbers, very similar ratios. Certainly not the same in every species, but the ratios between the adenine and thymine very similar in its traits. It's interesting, as humans we have more adenine and thymine than guanine and cytosine. But what this gentleman did, Chargaff, I'm not sure how to pronounce his name, he came up with these rules. And his rules are that the composition of DNA varies between species, so not all of us have the same genetic makeup, which we know now. And at this time, this was earth-shattering. It's ho-hum to us now, but it was earth-shattering then. And that many species, the number of A and T bases are equal, and the number of G and C bases are equal. This means that it was inferred that then there must be some a correlation between the A and T and the C and G. There's another piece before they totally understood the discovery of the double helix, but what happens now is Maurice Wilkins and Rosalind Franklin used a technique called crystallography to study the molecular structure. It's an X-ray technique called crystallography, and you actually have the image here next to Rosalind Franklin's her her portrait, I guess her official portrait, you actually have the image, and then that's her portrait again at the bottom. It was a picture that I found that I thought was kind of cool to have another look at this image. But you have this double helix, you have this binding in the middle, and the actual structure of what the DNA would look like. They produced a picture of DNA molecule using the technique. It was known then to Watson and Crick. One of the things, unfortunately, Watson and Crick didn't quite give as much glory to Franklin and Wilkins as they wanted to afterwards, after they, I guess, thought about things, because this image really helped them understand what the model may look like. It was a combination of this image, what Chargaff had learned about the proportions with regard to the nucleotide bases, and then understanding the bonding. What they did was they looked at different scenarios. If A bonded to A, which was what they thought at first, is that the same was going to bond together, they realized that they weren't going to get this uniform ladder. And if A bonded to C, this wasn't going to happen. So this is where you have this idea of the pyrimidines and the purines and the pyrimidines in terms of their molecular structure. And the fact that you have to match up a pyrimidine and a purine in order to have this regular ladder type arrangement, which they knew was the design of the double helix. The other thing is our old friends, the hydrogen bonds. You have triple hydrogen bonds between guanine and cytosine, and that's the most stable. If you have 3 bonding sites on one molecule and 3 bonding sites on another molecule, they're going to be the most stable if they stay together. And then on thymine and adenine, 2 hydrogen bonding sites. So that kind of locked the door in the fact that that's the arrangement with regard to the helix, is that structure is going to be designed that particular way. So we know it looks like a double helix. We know about its size at this point, we meaning the scientists. And we also know that it had this backbone, this sugar phosphate backbone, the nucleotide bases, and then Watson and Crick came up with the actual model of the double helix, the turning, and the understanding of what the DNA molecule looks like. The next thing they did was, how does it replicate? The next thing they wondered is, how does it replicate? So they built models. Franklin had concluded that the outer sugar phosphate backbones, that there were 2 of them, nucleotide bases. So it was kind of a combination of the 2 groups of scientists working together. And Franklin and Watson were in England, of course. I mean, there's always this rivalry between countries, and Watson and Crick were in the U.S. But they worked it out with regard to what it would look like, and the thing that's on this particular slide, which I should have moved ahead, is you have that word at the bottom, purine and pyrimidine, in terms of the pairing. And they realized that this has to be the pairing in order for there to be consistency in the width of the ladder. And here you have your purines and pyrimidines that when they pair up, not only in size but also in these hydrogen bonds, that Chargaff's rules are then respected. And so they definitely had the idea of what a DNA molecule looked like at that time. The relationship between the structure and function that made it possible for genetic material to actually code, because these nucleotide bases were repeats of the same 4 over and over again, but in different sequencing. So the next thing, as I said, they wanted to go on to is to figure out how does it replicate, because they know it's replicating and allowing the next generation to have an inherited data. So they now knew there were two strands, two complementary strands. Watson and Crick thought that one of those strands is going to act as a template, so that the DNA is going to unwind, and two daughter portions, I guess, of the DNA, you might say, are going to be built onto the template of the original unwinding. So what you have here is the parent molecule in the image, separation, and then two daughter versions of the DNA being built on the template of the original. And then, of course, every time something's moved forward, people have to come up with ideas of how does this happen. So Watson and Crick were pretty set in what's known as the semi-conservative model. And the semi-conservative is that you have the original, one part of the original and then a daughter. It's kind of the image that I just showed you, and then one part of the original and the daughter, so that with each of the replications you have one side is the older DNA and one side's the newer DNA each time it replicates. That's a semi-conservative. The conservative model is that the DNA unwinds, and then a whole new one is made, and it gets together, and then the original one then gets back together again. And then the dispersive model is that the DNA unwinds in little pieces, and the daughter cell is made in pieces and then put together. The theory that won out, based on experiments by Matthew and Mendelssohn and Franklin Stahl, which I'll let you know which one of these names you have to know, but mostly you just need to know the experiments. Supported the semi-conservative because what they did was used a heavy isotope and a lighter isotope. So they had heavy and lighter. Between the old portion was heavier, the new portion was lighter in weight. And of course, then they can centrifuge, and they can centrifuge based on weight. And what they found was in all of the new cells, you had a heavy and a light, a heavy and a light. And so the semi-conservative model was then accepted as the way that DNA replicates. And so that then moved that particular part along. Still understanding the actual how does it do it was still a mystery, and still a mystery to be discovered. One of the things that's kind of eye-opening is the speed with which this replication can take place. Your book talks about, and I don't think I've left it in any of these PowerPoints, but it talks about a human cell can do about 5 million in a day. Replications. I was just reading that, how fast it can happen, and the let's see if I can, I can't pull it up this quickly, I'm sorry, but the replication is mind-boggling how fast it is and how many times the DNA replicates without any mistakes. It's just mind-boggling. If I could be that perfect in everything that I do, I would be a robot or something. I would be something other than myself. So there are very few imperfections, and there actually is a safety mechanism with regard to those imperfections, and we'll look at that. So the fact that there are mutations is really very surprising, and we'll pick this up on the next one.